Okay, this is figure 642 in your text. Just to reemphasize here, figure 642 is demonstrating the 31 pairs of spinal nerves. And again, in the previous slide we were looking at the 12 pairs of cranial nerves. And even though the spinal nerves and the cranial nerves together all make up the 43 pairs of nerves that are part of the peripheral nervous system, it's important to understand that those cranial nerves that we looked at in the last slide, as I emphasized, do not exit through the spinal cord. They come out from the brain stem per se. These nerves we're looking at now, the 31 pairs of spinal nerves, are designated as spinal nerves simply because they do exit through the spinal cord. And if you look at this figure, you can see in an ascending fashion, going from the brain down, that there's 8 what are called cervical peripheral nerves or spinal nerves. There's 12 pairs of thoracic spinal nerves. There's 5 pairs of lumbar spinal nerves. And there's 5 pairs of sacral spinal nerves. And finally, there's a single pair of coccygeal nerves for a total of 31 pairs of spinal nerves. Now, you can go back and look at figure 641 that we already had discussed just to kind of rememorize how these nerves exit and enter the spinal cord, because it's important to learn about these things, because several chapters beyond this chapter 6 we'll be talking about, of course, muscle, endocrine systems. All these things in the periphery outside of the central nervous system are innervated by these nerves, and as you'll see, these nerves bring information from the central nervous system to all these different organs and muscles in the periphery, and of course the muscles, organs, endocrine system in the periphery send information as well back to the central nervous system. So you're at that point of your learning here where you need to understand how this transfer of information occurs, and understanding how these peripheral nerves, the spinal nerves, the 31 pairs of spinal nerves and the 12 pairs of cranial nerves, bring information from the CNS and return information to the CNS is going to become very important. Now, as you look at this figure 642, keep in mind as these nerves exit the spinal cord, they generally innervate tissue that are nearby, not long distances from where they exit the spinal cord. So, for example, the 8 cervical nerves generally have control of muscles and glands in the neck, shoulders, and arms, and plus they send information from the neck, shoulders, and arms to the central nervous system, also including the hands. The thoracic nerves are also innervate various aspects of the shoulder, but also the chest and the upper part of the abdominal cavity. The lumbar nerves, the 5 pairs of lumbar nerves, are associated with the lower part of the abdomen, like the hips and the legs. And finally, the 5 sacral nerves are associated with the genitals and the lower part of the digestive tract. So as you can imagine, if you knew absolutely nothing about the peripheral nervous system at this point, that when people receive, it's one of the reasons it's important to learn how these peripheral nerves, in this case the 31 pairs of spinal nerves, work and what they do. It helps you to understand better when you see a patient who's been in some type of traumatic accident and had their spinal cord severed. Wherever the spinal cord was severed, as you can imagine, from that point down is where they're going to have problems. So when people sever, say, their spinal cord around T12, everything below that, there's going to be problems in their ability to deal with the lower part of the abdomen, for example, to be able to walk, and also to be able to control their bowels, etc., etc. So you start to appreciate what these spinal nerves do when someone has a spinal cord accident and these nerves get severed. Now, you saw previously that the cranial nerves had mixed effects. Some of the cranial nerves were only afferent, bringing information from the periphery to the CNS. Some of the cranial nerves were only efferent, bringing information from the CNS to the peripheral nervous system. but a lot of the cranial nerves had both afferent and efferent effects. Well, the spinal nerves all have afferent and efferent fibers, unlike the cranial nerves. So the spinal nerves are constantly all of them are bringing information from the brain to the periphery and bringing information from the periphery to the brain. So these spinal nerves, now, if you read your textbook, you'll see that there's actually a further breakdown of this system of the spinal, and this breakdown involves the efferent component of these spinal nerves, and the efferent division of these spinal nerves of the peripheral nervous system is subdivided into a somatic nervous system and an autonomic nervous system. And probably most of you have heard certainly of the autonomic nervous system at this point, and we're going to learn something about that. And again, you're at a point in this textbook where some of this stuff that we're going to learn now is extremely important in order to understand what's going to happen later on in subsequent chapters, especially in terms of skeletal muscle. And really the simplest distinction, as your book describes and as all textbooks describe, between the somatic and the autonomic systems, and again, these are the subdivisions of the efferent component of the peripheral nervous system. Efferent component is broken down into the somatic and the autonomic systems, and the simplest distinction between these two is that the neurons that compose the somatic division are those neurons that innervate skeletal muscle. So you'll be dealing a lot with that when you get to the section of this textbook that deals with muscle fibers. The autonomic component of the efferent aspect of the peripheral nervous system innervates smooth muscle, cardiac muscle, a lot of the glands involved in the endocrine system, and neurons in the gastrointestinal tract that control contractions and secretions in the gastrointestinal tract. So we're going to start breaking this down now. And in this part of the text, we're going to look a little bit more at the autonomic nervous system, whereas as I mentioned earlier, later on you'll look more at the somatic component of the efferent component of the peripheral nervous system when you actually look at the chapter on skeletal muscle. So let's take a break here and go to the next slide. Okay, this is table 6.9 from your textbook, and it basically it's kind of a synopsis slide of what we were talking about more extensively in the previous slide. Again, showing the breakdown of the efferent component of the peripheral nervous system. Again, it's broken down into a somatic component and an autonomic component. Again, these are bringing information from the central nervous system to the periphery. And again, as you see here in this table, the somatic component brings information from the central nervous system to skeletal muscle, and importantly, this information comes in a single neuron that leaves the spinal cord and goes to the skeletal muscle. Again, as you'll see much more when you do the chapter on skeletal muscle. However, you'll see that this is very distinct from the autonomic system in a second. It only innervates skeletal muscle, as we said several times, and it only leads to excitation. In the case of muscle, that means contraction. So the somatic component here of the efferent division of the peripheral nervous system is a single neurons leaving the spinal cord and synapsing with skeletal muscle and causing contraction. The autonomic component here of the efferent aspect of the peripheral nervous system, as you're about to see, actually has a 2-neuron chain when it leaves the spinal cord and innervates the tissue in the periphery that it innervates, which, as I said earlier, is predominantly smooth muscle, cardiac muscle, glands, and the gastrointestinal tract. In the autonomic nervous system, also when it actually synapses with its target tissue, can either be excitatory to that tissue or inhibitory to it. So let's go now to the next figure, figure 6.43, and look a little more at this issue of having a single neuron communicating from the spinal cord with the periphery, as you have in the somatic component here of the efferent aspect of the peripheral nervous system versus this issue of having two neuron chains linked together that are actually communicating with the periphery in the autonomic nervous system. All right, let's go to that slide now and look at it. Okay, this figure 643, simply emphasizes what we alluded to earlier regarding the neurons that make up the somatic and the autonomic component of the efferent aspect of the peripheral nervous system. And as we discussed, and it's shown here, the somatic component of the efferent aspect of the peripheral nervous system, you have basically a single neuron leaving the spinal cord. It has its cell body in the spinal cord, and the neuron extends all the way to the skeletal muscle that it's going to innervate and releases a neurotransmitter and causes skeletal muscle contraction. It's only excitatory. The autonomic component of the efferent aspect of the peripheral nervous system is different, as we also alluded to earlier, in that you have a neuron leaving the spinal cord that's referred to as a preganglionic fiber because it synapses with a ganglion, thus the name preganglionic fiber. A ganglion is simply a group of neuronal cell bodies, as we'll see in a moment, that are outside of the central nervous system. So this preganglionic fiber synapses at the ganglion, releases a neurotransmitter which has some effect on the postganglionic fiber here, which in turn travels to the target tissue, smooth muscle of some type, cardiac muscle, glands, or gastrointestinal neurons. This aspect of the autonomic nervous system is extremely important in medicine. Particularly the neurotransmitters that are involved here. As you'll see shortly, the preganglionic fibers and the postganglionic fibers use different types of neurotransmitters. And furthermore, this aspect of the peripheral nervous system, this efferent aspect of the peripheral nervous system, the autonomic component, is further broken down into a parasympathetic and a sympathetic division. And probably most of you have heard those terms before, or learned about it in a biology class. But the autonomic nervous system is subdivided further into a parasympathetic division and a sympathetic division. And the neurotransmitters in these divisions there's many drugs that we use in medicine that either mimic these neurotransmitters, i.e. act as agonists, or antagonize these neurotransmitters for various reasons. Lots of medical implications for these types of drugs. And we'll be talking about these neurotransmitters in a moment. But in a nutshell, as you're looking at this particular figure here, in terms of the autonomic nervous system, whether we're talking about the sympathetic division or the parasympathetic division, the preganglionic fibers all use acetylcholine as a neurotransmitter. It's the postganglionic fibers that differ in terms of the neurotransmitter they use. Most of the time, it's either acetylcholine or norepinephrine. But that differs depending on whether you're talking about the sympathetic or the parasympathetic division. So think about that for a few minutes and then move on to the next figure, which is figure 644. And we'll start looking at this idea of a parasympathetic and a sympathetic component of the autonomic nervous system, where these nerves actually come out of the spinal cord, where their ganglia are located, and talk a little bit more about the tissues that they innervate and the neurotransmitters that they use. Okay, let's take a look at figure 644 here in your textbook, which this is basically showing the parasympathetic and the sympathetic divisions of the autonomic nervous system. And you guys had to spend a little time on this figure because it has a lot of information in it that's useful and will be important to you later on when we start looking at specific innervation of various types of organ systems, the heart, the bowels, etc. Now, one of the things that should stand out right away is where the preganglionic fibers from the parasympathetic and the sympathetic divisions of the autonomic nervous system leave the spinal cord. So the parasympathetic division on the left in this figure in the red lines, you can see the solid lines which represent the preganglionic fibers. You can see that these fibers leave the spinal cord as the cranial nerves 3, 7, 9, and 10. And they also exit the spinal cord as sacral nerves 2, 3, and 4. So there's no parasympathetic preganglionic fibers leaving the spinal cord in the cervical aspect or the thoracic aspect or the lumbar aspect of the spinal cord. So this is why the parasympathetic division of the autonomic nervous system is also sometimes referred to as the craniosacral division. Now, in contrast to that, if you look over on the other side at the sympathetic division of the autonomic nervous system, you can see that the preganglionic fibers, the solid blue lines, exit the spinal cord at vertebrae T1 through L2. So all of the thoracic vertebrae and lumbar vertebrae 1 and 2 have preganglionic sympathetic fibers exiting them. The other thing to notice, but there's a major difference here in this figure, is where these preganglionic fibers actually form synapses with the postganglionic fibers. And again, the postganglionic fibers for both divisions here are drawn as the dotted lines. And if you look on the right, again at the sympathetic division, you can see that the ganglia, which is where the cell bodies of the postganglionic fibers arise, are much closer to the spinal cord for the sympathetic division than they are for the parasympathetic division. In fact, you can see that in the case of the sympathetic division, they form two chains of ganglia that are referred to as sympathetic trunks. And if you look at these things, you can see that they are subdivided on the top, again looking at the sympathetic side, where anatomists have identified what are referred to as superior cervical ganglia, middle cervical ganglia, and an inferior cervical ganglia. And again, the preganglionic fibers that innervate these ganglia do not come from the cervical vertebrae. They come, if you look at this figure closely, from T1, T2, T3, and T4, and then they ascend upwards and they form these superior, middle, and inferior cervical ganglia. These ganglia are actually physically located very near the cervical vertebrae, but again, the preganglionic fibers that are part of these ganglia come from T1 through T4. If you look more again at the sympathetic side, you see that T5 through T10 you have preganglionic sympathetic fibers that descend to form what are called celiac ganglia and superior mesenteric ganglia. And subsequently you have postganglionic fibers leaving these ganglia that are involved with the innervation of the digestive system. Now, you'll talk a little bit more about this innervation of the digestive system, which is referred to as the enteric nervous system, when you get into chapter 15. You also have an inferior mesenteric ganglion, as you can see here on the right, where the sympathetic preganglionic fibers arise from L1 and L2. So the bottom line here is you have a parasympathetic and a sympathetic division of the autonomic nervous system. They all have preganglionic fibers leaving the spinal cord at different places, and they all form ganglia somewhere in the body, and that particular ganglia is much closer to the spinal cord in the case of the sympathetic division than it is in the parasympathetic division. The sympathetic division, by the way, is also sometimes referred to as the thoracolumbar division, just like the parasympathetic division is referred to as the craniosacral division, simply because of where the preganglionic fibers arrive from the spinal cord. As you're going to see in subsequent chapters, these systems, the autonomic nervous system, the two divisions, the parasympathetic and the sympathetic, tend to drive homeostatic function in opposite directions. In other words, the sympathetic division and the parasympathetic divisions of the autonomic nervous system innervate lots of tissues. And generally what happens is in those tissues, whatever aspect of that tissue function they're innervating, the sympathetic division drives it one way, and the parasympathetic division drives it the other way. On your figure here that you're looking at, you can see that they've listed here on this figure that on the sympathetic side, emergency responses, and on the parasympathetic side, they put rest and digest. That's certainly an oversimplification, but the reason they have that there is the sympathetic division certainly is more involved in the so-called flight or fight response. When that system is turned on, certain things happen that you need to happen if you're finding yourself in a potentially lethal situation. Just to give you some examples, for example, the pupils of your eye will dilate because you need to see clearly if you have to run for your life or fight for your life. Blood flow is shunted from peripheral areas like the skin to the muscle, where you're going to need it, obviously, if you have to fight or run. Bladder function or renal function is shut down because you don't need to be making urine, obviously, if you're running for your life. There's several things that are involved in flight or fight that are activated by the sympathetic division because it makes sense. There's things that need to happen if you're going to survive some type of serious problem that's confronting you at the moment. On the other hand, the parasympathetic division seems to be more involved with things that we're doing when we're not in an emergency situation, when we're resting and digesting. So the neurotransmitters that as I alluded to earlier, that are utilized by the sympathetic and the parasympathetic nervous system are almost exclusively acetylcholine and norepinephrine. In some subsequent figures here, particularly Figure 646, we'll look at that, and Table 610, talk a little bit more about these neurotransmitters and the type of receptors used, the type of acetylcholine receptors and the type of norepinephrine receptor. But what I want you to walk away from this figure with in terms of the issue of neurotransmitters, just remember all the preganglionic fibers here, whether they're sympathetic or parasympathetic, use acetylcholine as their neurotransmitter when they synapse with their ganglia. On the other hand, the postganglionic fibers that then travel from the ganglia to the tissue that's being innervated, if it's the parasympathetic division, the neurotransmitter that's the postganglionic neurotransmitter is also acetylcholine. On the other hand, in the sympathetic division, the postganglionic fiber neurotransmitter that's used is norepinephrine almost all the time. One exception I'm aware of is that sympathetic, postganglionic sympathetic neurons that are involved with sweating actually use acetylcholine as their postganglionic neurotransmitter. So that's the one exception to the rule that I'm aware of. But anyway, keep that in mind because it's going to be useful later on, this idea of preganglionic, postganglionic fibers, the neurotransmitters that are used, and as we'll see subsequently, the types of acetylcholine receptors that are involved and the types of norepinephrine receptors that are involved are extremely important because we make a lot of drugs that either act as agonists of these receptors or antagonists of these receptors. So spend a little more time on this, take a look at it, think about it, and when you're ready, go on to the next slide, which is figure 6.45. Okay, let's look at figure 645 here. This is just showing the sympathetic trunk that lies outside of the spinal cord. Of course, there's two of them on either side of the spinal cord, and there's only one drawn here for us to look at. Keep in mind also, if you've forgotten, refer back to figure 641 in this chapter if you've forgotten the basic anatomy of the spinal cord and the ventral and dorsal roots. As you're looking at this picture here, remember the ventral roots are where the ventral and dorsal roots come off the spinal cord and form the spinal nerves, and it's the ventral roots that brings efferent information or information from the central nervous system, and the dorsal component brings information or sensory information into the spinal cord. So looking at this figure here, 645, you can see the ventral roots containing the preganglionic sympathetic fibers is coming out of the spinal cord. It's drawn in blue. And remember, the sympathetic and parasympathetic systems, that component of the peripheral nervous system, is all efferent information. So it's coming out the dorsal root, and then it's synapsing on these ganglia, these spinal ganglia called sympathetic ganglia, again, that run up and down each side of the spinal cord. And it's synapsing with a postganglionic sympathetic neuron. Now, keep in mind, as you saw in the previous figure, that the actual sites where the preganglionic sympathetic fibers exit the spinal cord is only between T1 and T12, all the thoracic vertebrae, and L1 and L2. But these sympathetic trunks that run up and down the spinal cord go all the way up and down the spinal cord. They don't just run parallel to T1 to T12 and L1 and L2. So you can see it in the way this figure is drawn. You can see that some of these preganglionic sympathetic fibers, again drawn as the solid blue line here, come out on the ventral root and then travel up or down the sympathetic trunk before they synapse with their postganglionic fiber. This is also true for what are called the collateral ganglia, and those collateral ganglia are the ganglia that are involved predominantly with digestion. The celiac ganglia, superior mesenteric ganglia, and the inferior mesenteric ganglia, which you saw on figure 644. Those ganglia lie outside of the sympathetic trunks, and that's why they're referred to as collateral ganglia. But look at this and remind yourself of how information flows out of the spinal cord. And again, if you have to look back at figure 641, do that. But you're seeing here an example of efferent information coming from the sympathetic nervous system, leaving the spinal cord. You can see the cell body is in the gray matter of the spinal cord, exits the spinal cord through the ventral aspect of the spinal nerve, and then finds its post-ganglionic sympathetic fiber somewhere in the sympathetic trunk. Sometimes it has to travel up and down it a little ways to find it. Sometimes it's right there when it exits the spinal cord via the ventral root. The postganglionic fiber is right there. But just be aware of this fact in terms of physiology as well as anatomy, that this sympathetic trunk runs the length of the spinal cord, despite the fact that the sympathetic nerves only exit the spinal cord between T1, T12, and L1 and L2. Alright, at this point let's go on to the next slide. Okay, table 610 from your book shows the location of the receptors for acetylcholine, norepinephrine, and epinephrine. And again, acetylcholine and norepinephrine and epinephrine are the major neurotransmitters that are involved in the autonomic nervous system. Epinephrine, as you'll see in a moment, comes into play because of the issue of the adrenal medulla. So if you look at this table, you can see that there's two types of receptors listed here for acetylcholine broadly: nicotinic receptors A and muscarinic receptors B. If this was a pharmacology course, we'd be breaking those down even farther, but for our purposes here, we only need to know about nicotinic and muscarinic acetylcholine receptors. The nicotinic receptors really are ionotropic receptors, which we've learned about previously in this chapter, and the muscarinic receptors are more metabotropic or G protein linked receptors. And again, they got their names because the nicotinic receptors have a greater affinity for the substance nicotine. They like to bind nicotine. And the muscarinic acetylcholine receptors have a greater affinity for a substance called muscarine that you get from mussels. But what we want to recognize here for a physiology course is where these receptors are located. So if you looked on postganglionic neurons here in the autonomic nervous system, remember, whether you're talking about parasympathetic or sympathetic aspect of the autonomic nervous system, both components have a preganglionic fiber and a postganglionic fiber. So As we've discussed, all of the preganglionic neurons in the autonomic nervous system release acetylcholine. And what we know is that the postganglionic neurons, of course, have to respond to this acetylcholine, so they have acetylcholine receptors on them. And we know that most of these postganglionic acetylcholine receptors are of the nicotinic type. Other places that you find the nicotinic type of acetylcholine receptor is at the neuromuscular junction when we're talking about innervating skeletal muscle, but we'll see that later when we talk about innervation of skeletal muscle, which is not an autonomic component of the efferent aspect of the peripheral nervous system. That's the somatic component, so we'll talk about that later. And in addition, some central nervous system neurons in the brain use the nicotinic acetylcholine receptors to respond to acetylcholine. The second type, the muscarinic acetylcholine receptors, you find those on smooth muscle, cardiac muscle, on different types of glands, and also you find these on some central nervous system neurons as well in the brain. These things can be involved in memory and learning, and we'll learn more about the role that these muscarinic and nicotinic acetylcholine receptors play in the brain itself when we talk about things like memory in Chapter 8. But for now, we're in the peripheral nervous system, and where you find these muscarinic acetylcholine receptors is at most of the synapses of the postsynaptic neurons of the parasympathetic neurons themselves. Remember, the parasympathetic neurons use acetylcholine both presynaptically and they use acetylcholine postsynaptically when they actually innervate their tissue. Again, cardiac muscle, smooth muscle, etc. So keep that in mind. The other major type of neurotransmitter, as you know, that's involved with the autonomic nervous system are the catecholamines, norepinephrine particularly, but also epinephrine, as I alluded to earlier, we'll take note of when we talk about the adrenal medulla. The receptors for the catecholamines, norepinephrine and epinephrine, are also found on smooth muscle and cardiac muscle and on glands, and they're also found to some extent in the central nervous system. These receptors, again, if this was a pharmacology class, we'd be breaking these down to alpha-1, alpha-2 adrenergic receptors, and beta-1, beta-2, and beta-3 adrenergic receptors. In fact, we'll look at that just a little bit more specifically when we get to table 611 here in a few minutes. So look over table 610 here, and the thing I want you to get from this is just be aware that the major neurotransmitters that we're talking about here in the autonomic nervous system are acetylcholine, norepinephrine, and to a small extent epinephrine. And just remind yourself that acetylcholine is used as the neurotransmitter in all the preganglionic neurons, whether it's sympathetic or parasympathetic, and in the postganglionic neurons, it's acetylcholine is used again as the neurotransmitter in the parasympathetic division, and norepinephrine is used as the neurotransmitter in the postganglionic neurons in the sympathetic division, with at least one exception, the sympathetic input to sweat glands. So think about that and try to get that clear in your mind as you look at table 610, and then go ahead and go to the next slide. Okay, figure 646 from your text. Again, just to briefly review here, this is more of the same, what we've been learning, because this is a rather complicated and important issue, and we want to make sure that you are clear with this so that when we go to subsequent chapters, this issue of autonomic nervous system hopefully will be soundly ground into your mind. So again, as you look at this figure, remember we said earlier that the peripheral nervous system, like the central nervous system, has an afferent and an efferent component. And we said that the efferent component is subsequently broken down into a somatic component and an autonomic component. The autonomic component, again, is broken down into a parasympathetic and a sympathetic division. So at the top here, what you're looking at is the somatic component of the peripheral nervous system, where you have a neuron arising in the spinal cord, its cell body, traveling to skeletal muscle. And in this case, this neuron always releases acetylcholine, which binds predominantly to nicotinic acetylcholine receptors present on skeletal muscle and causes skeletal muscle to contract. So this somatic component of the efferent aspect of the peripheral nervous system is it's always excitatory, and it's also voluntary. As you know, you can control your ability to contract your skeletal muscles. You can move your arms and your legs if you want to. On the other hand, as you look down below this is looking at the autonomic component, again, the parasympathetic, which is divided into the parasympathetic and the sympathetic division. And as we know, in this component, cell bodies of neurons leave the spinal cord as well in the peripheral nervous system, but instead of going all the way to their target tissue, they first go to some type of ganglia, as we've described previously. So in terms of the both the sympathetic and the parasympathetic division, those preganglionic nerve fibers go and synapse at the ganglia with the postganglionic fiber, and the neurotransmitter that's released there is always acetylcholine in both the sympathetic and parasympathetic division. And the type of acetylcholine receptor that's present on the postganglionic neuron is almost always the nicotinic acetylcholine receptor again. Subsequently, if you're talking about the parasympathetic division, the postganglionic neuron in the parasympathetic division goes on and innervates tissue, and the innervation is smooth muscle, cardiac muscle, glands of the endocrine system, GI system, etc. But as we've been saying, in the parasympathetic system, the postganglionic fiber also uses acetylcholine as its neurotransmitter. But in this case, the acetylcholine neurotransmitter that's actually on the target tissue is of the muscarinic type. It's a muscarinic acetylcholine receptor most of the time. Now again, looking at this figure, if you go down to the second one from the bottom, you see again the example of a sympathetic aspect of the autonomic nervous system. You see the cell body arising from the spinal cord, goes a very short distance before it actually forms a synapse in a ganglia with a postganglionic neuron. Again, just like in the above situation, the neurotransmitter released here is acetylcholine, predominantly of the nicotinic type. But in this case, it's a sympathetic nervous system, so the postganglionic neuron leaves the ganglia, synapses with its target tissue, which again is predominantly smooth muscle, cardiac muscle, glands, etc. Only in this case, it releases norepinephrine as the neurotransmitter, which in turn binds to some type of noradrenergic receptor. And as I alluded to earlier, the predominant types of noradrenergic receptors are alpha 1, alpha 2, beta 1, beta 2, and beta 3. Finally, at the very bottom of this figure, you see an example of the adrenal medulla, which is the medullary or middle component of your adrenal gland, which your adrenal glands are paired glands that sit above your kidneys. In this case, it's a sympathetic system, but if you look at this, you'll see that when the neurotransmitter is released, which in this case is mostly epinephrine, it's not released into a synapse, it's released into the bloodstream. So again, looking at the bottom here, you see preganglionic neurons come from the spinal cord, and they enter into the adrenal medulla, where they synapse with cells, specialized cells, mostly what are called chromaffin cells, and they release acetylcholine just like other preganglionic neurons do in the sympathetic nervous system. Now, these chromaffin cells produce and release a lot of epinephrine as well as some norepinephrine. But it's different here. Again, if you look at the situation just above this part of the figure, the norepinephrine and the epinephrine being released here is released into the bloodstream as opposed to being released into a synapse. But most of the norepinephrine and the epinephrine that you find in the human bloodstream does in fact come from the adrenal medulla. And this epinephrine and norepinephrine that's released into the bloodstream is certainly capable of binding adrenergic receptors at various parts of your body, just like the norepinephrine that's released into postganglionic synapses in the sympathetic nervous system binds adrenergic receptors that are present on the tissue there, the smooth muscle, cardiac muscle, glands, etc. So this figure is kind of more of what we've been talking about all along, but hopefully if those of you that are visual learners, this will be helpful to you to be able to look at this. And again, when you're looking from the top to the bottom, the top is the somatic component of the efferent part of the peripheral nervous system, and the bottom one here, all the bottom stuff in this figure, is the autonomic component. The top of the bottom, the one that starts with on the left, shows the CNS. It shows a preganglionic fiber going way over to the right side. That's a parasympathetic fiber, and it's synapsing and using acetylcholine, both that it's in the ganglia as well as its postganglionic fiber is using acetylcholine as well, binding muscarinic acetylcholine receptors. And again, below that is an example of the sympathetic component of the autonomic nervous system, where the preganglionic neuron comes out, finds its ganglia very close nearby the spinal cord, uses acetylcholine as the neurotransmitter there, just like in the parasympathetic division, and then the postganglionic fiber goes to the target tissue, and in this case releases norepinephrine. And finally, again, the bottom one shows the example of the adrenal medulla, where it's releasing its neurotransmitter, predominantly norepinephrine and epinephrine, into the bloodstream. So look this over, think about it, because all of this stuff that we've been talking about relative to the autonomic nervous system, as I said several times now, will appear later in subsequent chapters, particularly the cardiovascular chapter. So get comfortable with it now. And in fact, after you've looked at this for a little bit, we'll go to the next slide, which is just it's a table, 6.11, and we'll look at some specifics in terms of what these neurotransmitters do when they're released postsynaptically. Table 611 from your text is titled Some of the Effects of Autonomic Nervous System Activity. I want you to look over this table, but not because I expect you to memorize this table for exam questions, but I just want the idea of what the autonomic nervous system does to try to come together a little bit here for you. If you look at this table, you look across the top, it shows on the left the effector organ, and then the receptor type next to that. And this receptor type is for the it's the receptor type for the postganglionic neuron in the sympathetic component of the autonomic nervous system, because if you look down through these receptor types, you see they're all alpha or beta adrenergic receptors. And remember, it's only the sympathetic nervous system that uses norepinephrine as its postganglionic neurotransmitter, and it's norepinephrine that binds these types of receptors, these alpha 1, alpha 2, beta 1, beta 2, and beta 3. Then you look to the right of that, to the right of receptor type, and you see the effect that's produced by the sympathetic nervous system when these receptors are activated. And then to the right of that, it says parasympathetic nervous system effect, and it shows what the parasympathetic nervous system does on the same system, whether it's heart muscle, heart rate, etc., whatever it is. Take note of the fact that the parasympathetic nervous system effect is mediated by muscarinic acetylcholine receptors, even though that's not listed here. Again, at this point you should realize that the parasympathetic system is using acetylcholine as its final neurotransmitter, as its postganglionic neurotransmitter, whereas I said the sympathetic nervous system, its postganglionic neurotransmitter is epinephrine. So anyways, you look at this, the idea that I want you to kind of get is that, as I alluded to a slide or two ago, some or a lot of physiology actually is mediated by this autonomic nervous system, and it results in the sympathetic and the parasympathetic nervous system opposing each other. As I said earlier, Sometimes the sympathetic nervous system drives some physiological parameter one way, and the parasympathetic system drives it the other way. And some examples of that can be seen here. I mean, you can look at, if you look down the list on the left, if you look at the heart, and then you look under the across to the right under sympathetic nervous system effect, and you see increased heart rate by beta 1 receptors, increased contractility by beta 1, beta 2 receptors. Then you look to the right of that, and you just see that the parasympathetic effect on that parameter is that the sympathetic nervous system is producing is exactly the opposite. So they oppose each other for the most part in the heart. On the other hand, if you look down a little farther on the left to the arterioles, you look under the sympathetic nervous system effect, and you can see that most of the time the effect on the constriction or dilation of arterioles is mediated almost completely by the sympathetic nervous system without any input from the parasympathetic. So again, you look at coronary arteries, and you can see that alpha 1 and alpha 2 receptors are present on coronary arteries, and when norepinephrine binds those on coronary arteries, they constrict. If beta 2 receptors are activated on coronary arteries, it dilates the coronary arteries, etc. And you go right down through there. Look at skeletal muscle. It shows alpha 1. It constricts when sympathetic nervous system is activated, i.e. when norepinephrine is released. If it binds alpha 1 receptors in skeletal muscle, it constricts the skeletal muscle arteries. If beta 2 adrenergic receptors are activated when norepinephrine is released by the sympathetic nervous system, it dilates skeletal muscle arterioles. And right on down the line, you can see that many of these effects, stomach effects, intestine effects, gallbladder effects, you have the sympathetic nervous system driving some aspect of it one way using these alpha 1, alpha 2, beta 1, beta 2, beta 3 receptors, and you have the parasympathetic nervous system driving it the other way using muscarinic acetylcholine receptors. But again, sometimes there is no opposition. I just use the example of arterioles. If you go farther down here, you see, if you look at the fat cells in the kidneys on the left-hand side, you see that there's effects by the sympathetic nervous system that are mediated by alpha 2, beta 3, beta 1 receptors. But if you look over under the parasympathetic nervous system effect, there is no parasympathetic nervous system effect on the kidneys or fat cells, or very little anyway. And finally, at the very bottom, you can look at the skin, and you can see that the muscles that cause the hair on your arms to stand erect, when the sympathetic nervous system is activated, if the norepinephrine binds the alpha 1 receptors on these muscles that are involved in making your hair stand on end, then the hair will stand on end. And you look to the right, and you see that there's no opposing effect by the parasympathetic nervous system. So the effect is produced only when the sympathetic nervous system is turned on, and there's no parasympathetic antagonism of it. So again, don't get too caught up in looking at this table in terms of trying to memorize this. If you go on to medical school or even to PA school, what you're going to end up having to memorize mostly is the way the drugs work that either act as agonists or antagonists to a lot of these receptors that you're seeing here, and the effect that they produce. Again, to give you a quick example of that, look under the lungs here as the effector organ on the left. Look under lungs, the bronchial muscle, and you can see that the beta 2 adrenergic receptor, when it's bound and activated by norepinephrine, produces relaxation of the bronchial muscles. Again, it can be bound by either norepinephrine or epinephrine. Epinephrine and norepinephrine have differing affinities for these different types of adrenergic receptors, the alpha and beta receptors. So just looking at that should tell you, if you knew nothing at all about the pharmacology of adrenergic receptors, if binding beta 2 receptors dilates or relaxes the smooth bronchial muscle in the lungs, well, it should make sense to you that a beta 2 agonist then would be a useful drug to treat somebody with who was having some type of bronchospasms and couldn't breathe, like somebody who was having an asthma attack. And in fact, that's the case. So again, just get the big picture here of what the sympathetic and parasympathetic systems are doing, and get an appreciation for how they affect so many different physiological systems, sometimes directly opposing each other, sometimes having effects in the system, in a physiological system by themselves without opposition from the opposing component of the autonomic system. Don't spend a lot of time memorizing specifics out of this table. So when you're done looking at this and thinking about it a little bit, let's go ahead and go to the next slide. Okay, let's take a look at this figure 647, which is the last figure we're going to look at in Chapter 6. As you look at this thing, try to visualize the layers that you'd have to go through if you were a neurosurgeon and you were drilling a hole through a patient's skull to access their brain. And if you did that, you would see that after drilling through the skull itself, you would first encounter one of the three types of meninges that cover the brain and the spinal cord. And this meninges is the dura mater. The dura mater, or the dura as some people call it, is simply the outermost covering of the brain and spinal cord. And it's the thickest and toughest of the three meninges. After you went through this, you would then bypass the large veins that run under some parts of the dura at the top of the head, and you'd contact the arachnoid mater, which is the second meninge. After penetrating that, the arachnoid mater, your drill would then pass through the subarachnoid space, which is just beneath the arachnoid mater, and you would likely be surrounded at that point by cerebrospinal fluid, or your drill would be anyway, because there is cerebrospinal fluid in the subarachnoid space. And just at the bottom of this cerebrospinal fluid, you'd see the very thin pia mater, which is the thinnest of the meninges, and this is the meninge that actually lies directly over the nervous tissue of the brain and the spinal cord itself. After you cut through that, you would actually see the brain and or see the spinal cord. The ventricles you see in this figure are simply spaces in the brain that hold cerebrospinal fluid. The cerebrospinal fluid itself is made by the ependymal cells that we talked about at the beginning of this chapter. They're one of the glial cells, and they're part of the choroid plexus that you can see in this figure that are in the lateral ventricles and the third and fourth ventricles. The cerebrospinal fluid is being made constantly by these ependymal cells, and it's being drained constantly from the central nervous system, predominantly through these one-way valves that are in the large veins at the top of the brain, and then they just enter into the cerebrospinal fluid just enters into the general circulation. And as you can see in this figure, the cerebrospinal fluid flows from the lateral ventricles and third ventricle down to the fourth ventricle, and from the fourth ventricle it empties into some structures that aren't on here that are called the cisterna magnum and the brainstem cisterns. And from these, it flows into the subarachnoid space, which surrounds both the brain and the spinal cord. So therefore, the cerebrospinal fluid cushions the brain and the spinal cord from potential trauma, since the brain and the spinal cord literally float in it. Now, passing the cerebrospinal fluid through the ventricles and ultimately draining it into the veins at the top of the brain is important, because the ventricles are capable of producing 400 to 600 milliliters of cerebrospinal fluid a day, and that's enough to fill the fluid spaces in the central nervous system 4 times over. So obviously, since it's being made constantly, it's important that it drains constantly, because so much is made every day, much more so than is needed. So there are clinical conditions where it doesn't drain properly, and you've probably heard of them. The common one is called hydrocephalus, or some people call it water on the brain, but it's really cerebrospinal fluid on the brain. And usually when you have what's called hydrocephalus, it's a condition where the cerebrospinal fluid can't drain from the ventricles for some reason. Usually there's an obstruction, such as a tumor. So as the ependymal cells keep making this stuff, the ventricles expand, and if you're a baby and this happens to you, the bones in your skull haven't fused yet, so your skull will actually expand or come apart because the sutures haven't fused yet, and you can see a baby with this condition and its head appears to be very large, which is actually a good thing because the skull is expanding to allow the brain to expand. In an adult, the skull can expand because the sutures are fused. So when this occurs in an adult, the brain, the ventricles are expanding and of course putting pressure on the walls of the brain, and it can cause all sorts of trauma to the brain, and it has to be fixed. And one way that surgeons fix it is they can actually put a shunt down into the ventricles, run it out of the top of the brain, but underneath the skull, or out through the skull, excuse me, and under the skin at the top of the head. It never is exposed to the outside. But the shunt, or the tube, is run under the skin, down behind the ear, all the way down across your chest and into your abdominal cavity. So this shunt that surgeons use literally shunts cerebral spinal fluid from your ventricles down to your abdominal cavity, so it can drain into there instead of draining the way it normally would do. But anyway, when you look over this figure and think a little bit about cerebral spinal fluid, the meninges, and how it's made, read a little bit about the blood-brain barrier. There's very little written about it here, but just read what they've written about the blood-brain barrier in this section, just a couple of paragraphs, and then move on to the last slide, which is the final slide in this chapter. It's kind of a summary slide. Okay, chapter 6 was a lot of work. It's around 30 pages. A lot of information, especially if you haven't been exposed to the nervous system before. So after you've gone through this chapter and you've listened to all my my sound modules, and you've gone through that at least once, read through the entire chapter at least one time, the thing to do now that this information has been installed in you is to go to each of the section summaries. Remember, chapter 6 had 4 sections. Go to the section summaries at the end of each section and read these section summaries and think about those, and look at the section review questions again for each of the 4 sections of this chapter 6. So section summaries and section review questions, and then at the very end of the chapter you have chapter 6 test questions. There's 10 of those, and you have 7 quantitative and thought questions for chapter 6. The answers to the test questions and the quantitative and thought questions are at the back of the book, but I'd highly advise you to try to do those yourself before you look up the answers. It'll help you in the long run, but that's the way I would approach this if I were you. After you've done that, you've reviewed these section summaries, and you've gone over the test questions, and you've gone over the quantitative and thought questions, and then looked up your answers to see if they are correct. It'll be time to move on to chapter 7, sensory physiology, where a lot of the stuff that you've spent learning in chapter 6 will be applied, and that's particularly true in chapter 8 as well. Alright, good luck, and I'll see you in chapter 7.